Happy Wine Wednesday, everyone. Oh, I forgot to take Holly and Cheryl off. Holly and Cheryl will be back in just a little bit. But for the first part of tonight, I wanted to introduce my special guest. No, actually, you guys, you know how I jump around. Happy Wine Wednesday. Hope that your tanks are doing amazing. I'm so excited about this guest. I'm jumping over myself. But I'm sipping on Chardonnay. Let me know in the comments what you're sipping on. And with that, I will say we're also not currently going to Instagram. So if you have Instagram friends, tell them to jump on over to YouTube, search Seahorse Whisper, join us. And with that, with all my rambling, I'm going to introduce Kenneth from Hydrospace. Hello, Kenneth. How you doing tonight?
I'm doing well. Hello to everybody.
All right. And guys, I'm going to ask my questions. Kevin, I saw your post. Yes, I'm fashionably late as usual. Sorry. But we are here. Hi, Tanya. Hi, Aqua Chem. And hi, Kevin. And hi, everybody else who's joining us. Shyla. You guys ask your questions in the comments. I will go back through them. I'm going to go through mine, of course, first. But then we will get to yours, I promise. So, Kenneth, let's just start off with what the heck is Hydrospace? How did it come to be? What do you guys do?
Sure. Hydrospace is my small company that manufactures microbial products at this point, specifically for the aquaculture industry, which would include the aquarium industry. And then hopefully sometime in the near future, we will expand into the horticulture industry.
Crazy. And it's family run, right? It's like, I mean,
It
it's
is.
all in-house.
So far, yeah, a lot of ultra micromanagement.
And what got you interested in doing this? Like what made you decide to start this?
I've been doing this sort of stuff all my life, really, for like probably almost 25 years, starting out at a lab breeding zebrafish at the Institute in Neuroscience at the University of Oregon. And my primary responsibility there was cultivating the live feed for the fish. And I liked that a lot more than I expected that I would. So when I moved on, I finished college. I got a degree in biology, like just a general degree with a focus on ecology, which you'll see throughout is kind of my main interest really in using these. I'm not a microbiologist. I'm more interested in the nutrient cycling and ecological engineering and sort of those sorts of things that these microbes do. But then I moved on from the university to also in Oregon, the aquarium science program in Newport. And there I further concentrated on this stuff, more so with phytoplankton and things like that. But then, yeah, that was kind of the course of my work for many years after, working in hatcheries, working with rotifers, things like that. And when I ended up in Colorado, I came here to work for LG Barn, who with that, actually helped them develop products. At that time, the only thing they sold were pods and phyto, and they were in a garage in Longmont. And yeah, very small at that time. They've obviously grown quite a bit since then. And one of the problems we were trying to solve when I first started working there was with their Tigriopus specifically and their copepods in general. Two issues really: bioremediation. They wanted to be able to recover the water that we used, feeding them your live feed. As many on here, I assume, grow live feed, and that's an issue. Just water pollution. That was a problem there as well. And then also supplying enough carotenoids to make the Tigriopus nice and red. And in researching those two things, I found an interesting passage in Julian Sprung's The Reef Aquarium and Charles Delpy's The Reef Aquarium Volume 3, where they mention these bacteria. And through further research, I found that they do both things. They're used for bioremediation, and they're actually really rich in carotenoids, including the really valuable ones like astaxanthin. So to make that long story short, as you know, LG Barn exploded and expanded, and that project got sidetracked. And then they eventually got too big for our britches up here, and they moved down to the Denver area, into kind of an industrial area I didn't want to live in. So I was mulling whether or not I wanted to move with them, and then decided, those bacteria we're looking at were incredibly interesting and potentially very useful. And we couldn't find a source for them anywhere. So I thought, well, there's the demand and potentially a lot of great uses for them. So I buckled down and founded
Did it yourself.
Hydrospace. Around like 2017, 18 is where I really started researching them, and then we launched in 2019.
Okay, and if guys in the audience, because you guys know I'm not scientific, I'm not good with biology. So if he says carotenoid and you're like, what's a carotenoid? Feel free to ask it. If you guys all know and it's just me, I'll do my research, right? But I do know you mentioned microbiome. Can you tell me why that's important in a captive environment and how that all fits in?
Yeah, so that's an area we're just really starting to breach, especially in the aquarium hobby. We've been looking at this more, I guess, maybe like 20 years, roughly, something like that in the wild. But thankfully, due to the availability of certain analytical equipment like eDNA analysis, stuff like that, where we can actually more easily determine what species of bacteria are in these different environments. So that's really exciting. And what it's really just supporting what we already kind of knew anyway, that bacteria and archaea are the foundation of every ecosystem, and their interactions, not just with the organisms above them on the food chain, so to speak, but with each other, are incredibly important for nutrient cycling and all ecology. So it's basically the foundation of everything we're working with when we build a captive environment. And yet people like aquarists and horticulturists and so forth really are just starting to better understand how these things work, and even more importantly, how to control and manipulate them.
Well, and I know for the seahorse keepers, breeders, et cetera, that are joining us tonight and watch this later, we're going to get into the suppression of the vibrio species, because that's what we all care about, of course. But basically, if I'm hearing you right, basically you're just showing us that in a captive environment, you need these bacteria that you don't realize you need. And so we got to build from the bottom up and make it more like the ocean in order for them to stay healthy and fight off things and etc. So let's see. Okay, so PNS ProBio, what is it? What's in it? Tell me about it. Tell me how that fits into what we're talking about.
Sure. PNS ProBio is our flagship product, the first product we developed. It's a monoculture of Rhodopseudomonas palustris. And Rhodopseudomonas is a widely used microbe in actually as a bioremediator and as a probiotic. It actually has appeared in some human probiotics.
Oh, wow.
Also for poultry and fish and shrimp, especially shrimp, for the reasons that we're going to get into later, control of vibrio. But basically, that was the one we were kind of by default, it was the easiest one to get. So that's the first one we worked with. So we used Rhodopseudomonas to develop PNS ProBio, which is this fella right here. He's a lot of people recognize the packaging before they recognize the name of the product.
Right.
But yeah, that one's kind of like our all-purpose product, because it is designed mainly. It could be freshwater salt because it's cultured in a freshwater medium, and it can be used for bioremediation. It can be put in foods, so it can be used as a probiotic, and yeah. And then we have specialized products on top of that. But
I'm sorry. So basically, because we're used to, when we talk probiotics, we're used to the products we've used before, and they can be used as in feeds, as you discussed, and also being dosed to the tank. Is that how this product works? Same difference? You can dose it to the tank, make the water better. Just putting it very simply. You say it so
Yeah.
eloquently, but for people like me, I'm like, so what does it really do? It, instead of all the names, it's like it works. It helps this and it helps that. So,
Mhm.
but no, I got you. And real
It's
quick, I
really
am
that
gonna,
simple. I mean, you can
yeah.
just you can put it in the water or in the food. You can't overdose. It never goes pathogenic or it never produces harmful metabolites.
So
So,
it does. I know we talked with a guest a few weeks ago about how some bacteria can turn from, and correct me if I'm wrong. I should bring the others in. I will shortly, you guys. But about how bacteria that is nitrifying can turn into the sludge eaters. I can't remember the name of it. But so this kind does not do that.
actually, nitrifiers, the most familiar ones anyway, are autotrophic, meaning their source of carbon
That's
is
it.
inorganic. So they use
Mhm.
CO2 like a plant. They are not sludge eaters for that reason. They can't. They
They
can't.
can't turn. Okay. I
No, they
probably
can't
got it wrong
consume
then.
organic matter. These bacteria are actually Rhodosudomonas, the one I just showed you in PNS ProBio. That organism is widely regarded by biologists as one of the most versatile organisms on Earth. It actually can perform autotrophy, so it can use inorganic sources of carbon like CO2, but it prefers
Mhm.
organic sources. So that would be a sludge eater. And interestingly enough, in addition to being a phototroph or a heterotroph,
Heterotroph, yeah.
it's also a phototroph. So it prefers to get its energy from light. It's capable of performing photosynthesis, but its source of carbon for building blocks to create biomass are organic. So it's a photoheterotroph, which is pretty unusual.
Holy moly, you just blew my mind. And so, I mean, basically, that's why you can't overdose it, is because if you put it in your system or you're using it in your feed, then it goes to work in this area. But if the things it needs aren't available, it goes to work in another area. It will still do whatever it needs to do and
Mhm.
not become a hindrance. I gotcha. All right. Don't you guys love it when I keep it simple? Everybody else is probably like, Kelly, we all know this stuff. Shut up, but I don't. So I like to ask the questions. And I know we've kind of already covered it, but just in general, some people are like, don't really understand probiotics. So why are they so important in a captive environment? And like in general, why are probiotics good?
In the case of these bacteria, I should mention purple non-sulfur bacteria, hence PNS in the PNS probio. Um, they were named after the purple sulfur bacteria, so they get a stupid name, purple non-sulfur bacteria. But anyway, the most popular one is Rhodosudomonas,
Right.
and that's actually as a probiotic. To answer your question, Kelly, is actually two things. One is it aids um, feed conversion. So when it enters the gut, it actually will consume things like cellulose that the animal cannot. It'll utilize cellulose as a carbon source. And in so doing, create biomass that the animal can digest along with vitamins and even antibiotic substances, which again, we'll get into later with the vibrio thing.
Yeah.
But then that's the other half. So feed conversion, and then also it inhibits the growth of pathogens, especially in the gut.
And that is so key for seahorse keepers. I know Cheryl's probably out there going, "Come on, let me in." She wants to ask questions
Mhm.
because that is like a game changer for us. Kevin asked real quick, "What's the difference between probio and substrate sauce?" I do want you to be able to talk about your other products. We do have people that aren't seahorse keepers that come. So, what's
Yeah,
the difference?
substrate sauce was so a lot of people wanted after PNS ProBio came out, a lot of people wanted to use it for cycling, and you can, but uh, we made a few modifications to make it more effective for that specific application. Since most of those people were saltwater aquarists, we culture the bacteria in substrate sauce in salt water, and then that just shortens the time it takes for them to become active. So, you
Gotcha.
know, which is important to people when they're cycling a tank. And then the other thing is the nitrogen source. We use ammonia as the nitrogen source. So out of the bottle, they're actually adapted to utilize ammonia. Again, more effective for cycling. And then I guess the third thing is, by the time substrate sauce came out, we started cultivating our second species, Rhodospirillum rubrum, another purple non-sulfur bacteria. So that one made an appearance in substrate sauce. So that one's a little bit better for diversity.
Mm-hmm.
2 species, so twice as much diversity.
No
But
kidding. Go ahead. Sorry.
yeah, but that's about it. They're about the same, and if they persist in the tank, they do the same thing. Once they can be cultivated on different media, but when they're in the tank, all subsequent generations, it doesn't matter. They're going to adapt to that environment. But the idea, in short, behind substrate sauce was to have something that behaved in a very specific manner for cycling right out of the bottle, which makes it work better, faster.
And just to back up, it's purple what? Non-sulfuric?
Purple non-sulfur bacteria.
And where did that come from? You said it came from purple sulfuric bacteria, right? What can you just tell us briefly what that's about? Because I would have no idea.
Yeah, they're not widely known because they're just not something we encounter a lot. They live deep in anaerobic substrates, so we don't see them. I guess a lot of us have seen them in high school biology class, maybe making something called a Winogradsky column, basically a tall, clear column that you pack with mud, and you'd have an organic source of carbon like paper that you'd wad up at the bottom. And over time, all these bacteria would grow, but they'd grow according to certain niches. They'd create like levels or layers in the mud. And you'd actually see that because most of these are pigmented. So the purple sulfur bacteria appear in a Winogradsky column somewhere in the middle. They're on top of the sulfate reducing bacteria, which are the ones that make hydrogen sulfide, which we all hate for its toxicity and rotten egg smell. The black stuff, if you're walking in really deep mud and that black stuff comes up, that stinks. Purple sulfur bacteria actually utilize that metabolically. Wow. So they help get rid of it, which is nice. So then the purple non-sulfur bacteria, which they're similar because they're also purple colored phototrophic bacteria, and they're sort of related. But the purple non-sulfurs were just discovered after the fact. So they're like, well, and like most organisms, they're a lot more sensitive to hydrogen sulfide. So they can utilize a little bit of it, but not much. Like purple sulfur bacteria love it. Purple non-sulfur bacteria, it can be toxic at certain levels. So hence being discovered after the fact, they were just called purple non-sulfur bacteria. But anyway, so they live right above the purple sulfur bacteria in a Winogradsky column, just below the surface of the sand bed. So they receive a little bit of light to perform photosynthesis, but they're still deep enough to where it's anaerobic or microaerobic. And then right on top of them is cyanobacteria and algae and stuff like that.
So it's anaerobic though.
They prefer anaerobic. So that's another difference, I guess. The purple non-sulfurs can tolerate or even grow in oxygen. So they prefer an anaerobic environment. But the purple sulfur bacteria, which are used to living deeper down, they're actually obligate anaerobes. So oxygen will kill them.
So wait, now you totally confused me. How does that work? How are they in our system? I mean, how do you use that in a system if it can't handle oxygen?
So our bacteria are purple non-sulfur.
Okay, sorry. No, I got it now. Sorry.
Yeah, that's fine. I know, the weird names, right? What's the point of that? But
and science and wine don't often mix. So guys, sorry if I ask stupid questions. But okay, Kevin wants to know what percentage of Rhodopseudomonas is preferred. I probably said that wrong.
Preferred?
Yes, I don't know what that. What do you mean is preferred? Kevin, let us know what you mean by that because I'm not sure. He asked it a while ago. That's the problem with these
They can
live
be,
streams.
if he's talking like percentage in an environment, so they're ubiquitous in the wild. I mean, they are found everywhere from forest soils to deep sea sediments. They've been isolated from the human belly button. But depending on the environment depends on how well they grow. Unlike in a coral reef, I saw one study where they were, I think, like number 6 in the top 18 genera, Rhodosudomonas was. And I mean, there's probably hundreds or thousands of genera occurring in a coral reef in any one site. So obviously, pretty important ecologically and definitely pretty abundant. They didn't give a percentage, but I imagine they're way up there.
And he did say in a microbiome sense.
Oh,
So
right. Yeah, so they're not, that's one thing you'll see in the literature is they're not necessarily always really abundant, especially in like in a well-established ecosystem. What they're really good at is you'll see them in larger percentages in a disturbed environment. So these are what they call engineer species in ecology. They go into a disturbed environment, or say like a new aquarium would be like a clean slate, I guess, like a disturbed environment. And they go in and they're good colonizers. So because they're so adaptive. So they'll basically make a disturbed environment, or let's say a new aquarium, more hospitable for other more specialized species. So they actually promote diversity. So you'll see a higher you'll see a higher percentage of them occurring in a fresh environment, so to speak, and then they may actually be less abundant down the road as other microbes come in and push them out. They're more restricted. They'll tolerate the presence of oxygen, but they eventually get pushed back into their real special niche of that little Goldilocks zone where there's light, but it's anaerobic and there's some availability of organic carbon. Like right under a sand bed.
gotcha. And so, and you discussed the new tank. So in our seahorse tank, say we add it, it's going to do its job, and then what? What happens to it? Did that make sense?
Yeah, well, it depends on the tank, but in situations where you have really high phosphate and nitrate, you might see a reduction in nutrients and therefore a possible reduction in algae, depending on how the tank is filtered.
Sure.
So, for people using a skimmer, you'll see a lot more export of the bacteria. I should mention, even though they live in an anaerobic environment and form biofilms there, the daughter cells, the new cells, are motile, so they swim around, and that's when they're susceptible. So if you have corals, corals will eat them. Corals love them, actually, both in aquariums and in the wild. But when they're swimming around, that's when they can get removed by a skimmer. So they're like a means of nutrient export where you don't have to dose carbon, if that makes sense. They can actually use carbon that's in the water that other bacteria can't use, like cellulose from dead algae. So they'd be particularly useful like in a seahorse tank where people have a lot of macroalgae, because macroalgae release a lot of yellowing compounds into the water, different tannins and polyphenols of different kinds. And these bacteria can actually utilize them as a carbon source.
Oh,
And
man.
what's important is a lot of other bacteria cannot. They don't possess the enzymes to degrade those substances. So these bacteria would grow really well in an environment like that anyway. And my point is, a lot of seahorse people tend to have a lot of macroalgae. So that's just one example of where the special abilities of these bacteria to utilize carbon might be useful.
We talk all the time about how we create these extremely sterile tanks. And when I bring Holly in, we'll talk more about how we kind of wonder if that's really that good. And so I like the fact, I mean, we still are promoting creating the sterile tank, but it would be great to be able to add in what you want to add in to work in the way you want it to work, like this create the natural the natural setting without, using live rock or whatnot that brings in pests, because that's the big fear with
Oh
seahorses.
yeah. I
And
don't.
we get
Mhm.
criticized a lot because you don't have a natural setting. And so are they really getting what they need? But the whole point is make it sterile, then put in what you need. And you're
Right.
allowing us to do that. I love it.
And think about
Um,
the people carbon dosing. You're not
yeah,
just promoting the growth of the beneficial microbes that you're targeting to remove the nitrate and phosphate, but you could potentially be
growing the bad
promoting
guys.
the growth of other heterotrophs, including vibrio. So
Yep.
it's a very risky carbon dosing, but in this case, as you pointed out, you're only adding the bacteria that you want, dosing the
Exactly.
bacteria rather than.
Sorry, dropped my phone. Okay, so two things I want to say real quick is for any of you reef keepers out there that are with us or watching later, Kenneth went on Tank Talk with Tyann, and he talked about all the benefits to a reef tank. There are multiple that go beyond this expanse of what we're going to discuss tonight. You need to go watch Tank Talk with Tyann. It is still on YouTube if you can't find it in the Facebook group. But they talked a great deal about how this bacteria promotes foods for the zoo, and I can never say that word, zooxanthellae. How you say that? Say it for me. Anyways, go ahead.
Probably at least two different ways I say it. So
Yeah. So my point is, if there's questions like that, we will ask them and at the end and let Kenneth answer them. But you can always go watch Tank Talk with Tyann. But for tonight, for us Seahorse people, I do want to just dive right in. And I know. Oh, the second thing I wanted to say is Kenneth actually made us a notes that I will be posting later and be making available to any of you to talk more about Vibrio and what's going on with Vibrio and how this product affects Vibrio and why we want to do it and all of that. But I'm going to let you dive right in. Kenneth, what is Vibrio? Why do we hate it? And how does your product stop it or suppress it?
yeah,
Excuse me, not stop it.
here's where I grab the notes.
Yeah,
Printed out, old fashioned like. Um,
I love it.
so you
I've been looking at notes all night. You guys know me, but go ahead.
so you guys
Sorry.
know from much more familiar with vibriosis than a lot of other people because it's so prevalent in your niche and so devastating to the animals you try to keep. Vibrio attacks all kinds of organisms, but including fish, but in all the species that affect seahorses. But I was even looking where they just discovered a specific strain in a seahorse population, I think off the coast of Spain, and it may become a new species, one that may actually specifically target seahorses or even that population of seahorses. So anyway, for whatever reason, seahorses it's devastating, and fortunately,
Cheryl will cue you into why it's so devastating when she gets on. But go on.
yeah.
Sorry.
So and fortunately for you guys, there's actually a growing trade in the culture of seahorses, because in China, as a lot of you know, for better or for worse, they are used in medicine
Medicinal.
and things like that, even the curio trade. So the demand has outpaced the supply, and now they're actually culturing a lot of seahorses and encountering vibrio. So it's become a problem that money is worth solving now. So they're focusing research on that. And if you go online, you'll find a lot of papers on
Yep.
seahorse
Dan's
vibriosis.
talked about that a lot. Sorry, go
Yeah,
ahead.
sure. So, I know that. So, like, we're going to be talking. So, just for those who there may be some newer seahorse keepers on here, and we may be using terms like vibriosis, flesh erosion disease. And I'm not a seahorse person, so you can tell me if I'm wrong on any of these. But these are commonly used for seahorse vibriosis, marine ulcer disease. And then there are terms like flesh-eating bacteria, ulcerative dermatitis, hemorrhagic septicemia. Are those all terms familiar to you guys?
To me and to those that like Cheryl, of course,
Okay.
absolutely. But go ahead. I mean, if you want to expand upon it, please do.
Yeah. So a lot of things like sudden death syndrome, for example, are maybe caused by Vibrio, but it's important to note that a lot of these diseases, we may see symptoms that could also be ascribed to other pathogens. Some of them aren't even bacteria. So things like Pseudomonas, Mycobacterium, Urinema. And it's just important to note that so we don't.
Our seahorse people were very guilty of automatically jumping to its Vibrio. And it's not always. Cheryl points out all the time. Could be Myco. You're absolutely right. But go on. I'm sorry. I didn't
Sure. And it just because at the peril of the animal, because you could actually end up using the wrong treatment. But vibriosis obviously is caused by Vibrio. You're going to come across in seahorse land Vibrio harveyi, Vibrio algolinicus, Vibrio splendidus, and I only saw it in one case or one citation, Vibrio tubiacei. So there's 14 clades consisting of like 86 different species, and I've seen that Vibrio in general can attack seahorses. So there obviously are a lot of different species and even different strains within those species. So treatments may vary depending on in some cases, some strains of Vibrio are now resistant to certain antibiotics, for example.
Oh, I know it. Ugh.
Yeah, right.
Go on.
So
Sorry.
no, and that's actually an interesting thing. Before I dive deep into this, just some other stuff I saw that I thought was cool, or concerning anyway, was because of the wide use of antibiotics. There are populations of seahorses in the wild now that have been exposed, and actually it's changed the microbiome. And actually altered the microbiota in their gut and in the pouch, and that's having health consequences for wild seahorses with respect to different diseases. So, once
I got to hook you up with with Chris Carey and with Graham Short. I got to hook you up with these guys because they need your help. But go on. Sorry.
So just Vibrio, just real quick, like they're proteobacteria, like these bacteria, actually really ancient heterotrophs, and they are flagellated, so they can swim around. They get from seahorse to seahorse very easily.
And they're omnipresent, so they're going to be in the tank.
In both natural,
in
both natural and captive environments. So they're always there, but pathogenesis is usually brought on by certain conditions. So even though they're always there, it's things like heat stress is a big one. Right?
That happened to me literally, and Kevin said, what caused the population of Vibrio? Saltwater natural occurrence. He's getting ready to answer that, but heat happened to me. I'll tell you later. Go on. Sorry.
Yeah. Well, with heat stress, obviously it stresses many, especially the temperate species of seahorses. It even going like a few degrees out of bounds and at the upper end will denature some of the components of their immune system and actually take away their ability to fight Vibrio. And on the other hand, it affects Vibrio itself by promoting their growth. So Vibrio grow faster when it gets really warm, and it triggers something that actually encourages them to become virulent. So it makes them more. It actually turns them into disease agents, I guess, the heat. So, and then I guess other things like poor nutrition, poor water quality, biotic stress. So if they're getting picked on by other fish, getting stung by a coral or an anemone or something like that in the tank, that will also, just by virtue of stressing the animal, generally compromise their immune system, and they'll be more susceptible to vibriosis.
Yep.
So you guys are probably familiar with the symptoms, but I would run through them since some of them are symptoms of other diseases. So the depressed appetite is one we see early, and then eventually you'll see, still early, reddish inflamed patches over the body. Those will turn into open wounds with some skin rot or skin sloughing. That's as it becomes more advanced. Not to be confused with heater burn. Sometimes those symptoms apparently look like vibriosis if a seahorse attaches to a heater and gets burned. Some probably a lot of you guys already know about, but worth mention. And then tail and snout rot can be caused by vibrio, but it can be caused by other bacteria as well. So that might be a tough one, or one to be very look for very specific symptoms before you set up any kind of treatment. And then,
well, Holly's going to want to talk to you about that stuff too, because we've had some confusion, when you don't know what's going on, how do you treat? But I'm sorry, I keep interrupting you. I apologize. I'll tell my story about my heat-induced vibrio situation later. Okay. Go on, sir.
And I'll try to blast through this and hit everything, but there are a couple odd ones really that I thought. Swelling around the eyes in the seahorse world, they call that donut eyes.
Yep.
That's indicative generally of vibriosis. And then you can get some bloating in a really advanced case. That's usually due to internal infections, the liver, kidney. And then they can get blood infections, which is really bad. That one you usually don't see much on the outside, but they get extremely lethargic and have difficulty breathing. And they say that's pretty much a death sentence, a blood infection of vibrio. So that's what I got for symptoms. And if you had any questions before I move on to treatments.
Yes, please talk treatments and then get it back into how you can
help.
Yeah,
exactly. Yeah. So I want to just hit the things people usually do. So there are antibiotics. I'll just run through a list. You can tell me how many of these ring a bell, but tetracycline, gentamicin, doxycycline, sulfonamide, metronidazole. Then the ones I would be more interested in with these bacteria, which are kanamycin, neomycin, and streptomycin. And then there are also some immunostimulants like beta-glucan, which you guys may be familiar with, and some antibacterial agents that are commonly used, such as chloramphenicol. I think you know Furan too from a video I saw.
Yes, actually, I am going to interrupt you one more time. Zen Ginger, thank you for posting the video. Actually, I show all of the symptoms that he just described in that video happening to my seahorse due to a heat increase that wasn't under my control. And now, and I was able to use Furon too, which is no longer available.
Oh,
so
really?
Go on, Kenneth. Yeah. I'm sorry. Yeah,
no, that's fine. So they're the antibacterial agents we just mentioned. And a weird one that I thought was cool is bacteriophages. They use some that are known as vibriophages, which are, so a phage is like a virus that specifically attacks bacteria. And there you can get some that specifically attack vibrio, which
really?
Yeah, which is pretty cool. I don't know how well that's been used in aquaculture or anything like that, but it's worthy of mention for someone looking forward to see, if any of these things ever become available in the hobby. And then finally, probiotics, which is my domain. So that would be organisms such as Rhodopseudomonas, which I mentioned, and at least one other one that we cultivate, Rhodobacter spheroides. They do this through competitive exclusion. So when you, either in the water or in their food, that means competitive exclusion is where they,
outcompete.
That could be through exploitation competition, where they outcompete the competitor for space or food, like nutrients. And then there's also interference competition, and that's something you see more when you actually use a probiotic that secretes antibiotics. So they're actively secreting compounds that kill the competitor. Yeah. So purple non-sulfur bacteria, such as Rhodopseudomonas, have been investigated particularly because of their carotenoids, which, because you asked earlier, those are, they're just like pigments. So everything from like lycopene, which makes tomatoes red, to beta carotene, which I'm assuming make carrots orange. And they're antioxidants. They do all kinds of good stuff. But it turns out,
well, you mentioned one earlier. You mentioned a specific product earlier that we know very well. I can't think of it now, but to turn seahorses red or to turn
astaxanthin.
clownfish red. That's it.
Yeah.
Right. Go on.
An interesting thing about astaxanthin, if you're using that for fish, just as a side note, if you're using, so one of the hatcheries I worked at, our supplier for astaxanthin changed the formula without telling us. Originally, it was, I think it originated from an algae. So it was a biologically sourced, it was a biological source. And when they switched it, unbeknownst to us, in this case, it was clownfish. They went from the bright red, which we typically were well known for, to like this icky yellowy orange color. And we couldn't figure out why it was. And then we finally narrowed it down to this source of astaxanthin. They had changed it to a synthesized source.
Oh, no.
But it wasn't exactly the same structure chemically. Right. It was chiral, which means it's kind of like your left or right hand. It's the exact same thing. It's a mirror, but they don't fit, right? Because your left and right hand can't be. They're still different. Left from a left and right perspective. So anyway, that was different enough to prevent the fish from being able to take it up. And what that proved to us is that astaxanthin absolutely does affect the coloration of fish, and even its specific form is really important. Another cool thing about astaxanthin is that I believe it's fat soluble, so it has to be, you can't just put it in anything and the fish can take it up. It has to be taken in with food, especially lipids. So a whole cell source of astaxanthin, like an algae or a bacterium, is vastly superior to just getting a powder, something like that, and mixing it in your food. Yes.
Cheryl's in the background doing cartwheels. She loves these words, lipids and astaxanthin, and yes, you're talking our language. Go on, sir.
Yeah. Sorry. So to continue with where we were going, the carotenoids, it turns out, that are present in Rhodopseudomonas, that's what gives them their purpley red color, and other purple non-sulfur bacteria. They're rich in astaxanthin, and these particular carotenoids, or at least some of them, have antimicrobial properties that are specifically shown to inhibit vibrio, at least in the case of Rhodobacter, which is another bacteria, another species we cultivate, which was really exciting. So now we have one way, actually two ways, that these bacteria, when used as probiotics, can inhibit vibrio. One, through exploitation competition, where they're just for space and nutrients and so forth, but also through interference competition, where they're secreting antibiotics and carotenoids. So the antibiotics, so Rhodopseudomonas, the one that's in PNS ProBio, is capable of synthesizing gentamicin, kanamycin, and streptomycin. So
what? Yeah. I mean, seriously? Go on. Sorry.
So it's a natural source of so a probiotic is a natural source of antibiotics. And what's really cool about that is it may limit its competitor's ability to become resistant. So say
that part confuses me. I was going to say, if it's releasing this, we always talk about, and you talked about, you don't want to use antibiotics in a limited fashion because then they become resistant. So I'll shut up and let you talk.
And that certainly may occur if we're putting it in their food. But it just seems like if you're putting it in the water all the time, that it's less targeted, and then there are other populations in the aquarium, say, right? So if you're putting it in the food,
hit with it.
yeah, so then other organisms could potentially become resistant, or other strains of the same bacteria or whatever. So yes, it's worth pointing out that if you're putting it in the food and you're doing that all the time, then organisms in the animal's gut in that moment could develop a resistance, but certainly less so than if you're just putting it in the water willy-nilly, where everything's getting exposed to it and can develop resistance outside of the gut. One thing about these particular antibiotics, if there are lots of antibiotics that will inhibit vibrio, but the particular ones that I looked up for Rhodopseudomonas, I believe, are all permeable through the intestinal tract. But some are not. You either you're forced to put it in, use it in different ways, put it in the water and just hope you kill it in the water, put some kind of substance that binds it or whatever. I'm not
focus. Yep.
Things like that get it through the gut. So it's kind of cool in that way. But yeah, so this is actually something that's been used already in the aquaculture industry. So shrimp, Pacific white shrimp, they're particularly susceptible to Vibrio hemolyticus, and it was devastating to the industry. It's actually a disease where Vibrio specifically attack the shrimp's liver. Found that using these bacteria in the water and in the food actually reduced mortality of these farmed shrimp. And then they went on to show that Rhodopseudomonas clearly inhibits Vibrio on the Petri dish. So, wow. Yeah, you can specifically see that, what they call a zone of inhibition on a Petri dish, where you put the two together and you see the one expand at the expense of the other. That could be there's probably several factors, and some of them are unknown. These bacteria are very complex in their interactions with each other. There may be lots of different compounds involved aside from the carotenoids and antibiotics. But what we do know very clearly is that Rhodopseudomonas inhibits Vibrio. That much is undisputed at this point. So now they're looking at that in fish, especially in the early larval stages, using it in first feeds and things like that, which again is something that would probably be really applicable to the seahorse world because of how often you guys use live feeds.
Right. Yep. And oh gosh, sorry, you blew my mind, and now I'm like looking through these comments and I got distracted. But someone did ask, are these bacteria affected by temperature? Like, tell us about that.
Temperature, yeah. So these bacteria won't even grow in low temperatures. So that could be an issue for, say, temperate seahorses. These bacteria really don't grow super well. Now, that doesn't mean they don't survive. They may be biologically active and capable of inhibiting Vibrio, even if they're not proliferating. But I would point out that they don't proliferate very well below, like, say, 75 degrees Celsius. So for some of your cooler water seahorses, that could be an issue.
What is 75 degrees Celsius in Fahrenheit?
Fahrenheit. I'm sorry. Fahrenheit. Yeah, I'm always trying to go back and forth. I know the optimal temperature for these bacteria is like 30 degrees Celsius, which I think is something like 83 degrees Fahrenheit.
So our seahorse tanks have to be 74 or lower.
Yeah.
Lower. Sorry.
Yeah.
So we're not pushing it though. This would still be okay.
Yeah. So the Vibrio and Rhodopseudomonas both like it warmer. So you'll see them both.
Well, that's the reason we keep our tanks cooler.
Yeah.
Go on. Sorry.
Yeah, but say if you have issues because if your vibriosis is coming on because of heat stress, that same heat would also promote the growth of the beneficial bacteria if they were added. So it's kind of a countermeasure in that specific scenario.
So you could possibly even keep like one of the things that many Cheryl and Holly are going to jump me when they get in here. But one of the things that many people want to do is keep seahorses in a reef tank setting. We can't keep them with certain fish. We can't keep them right. I know. Close your eyes. But I'm saying, when we talk just about coral, non-stinging coral, if I could keep my seahorses with coral that I love, and the only reason I can't right now is because of temperature differentiation, then if like these probiotics truly worked the way we say, out competing and etc., maybe you could. I mean, I don't know. I'm not telling anybody to do that, but I'm just saying
I would just say,
there's
like in the
thoughts for
case
the
of
future.
seahorses, where especially the temperate species, that would tilt the effectiveness of these bacteria when comparatively between like just general water dispersal or target specifically feeding them by adding it to their food. That would tilt it in favor of putting it in their food, because that way you're getting a good amount of them in the animal's gut. And even if obviously they're ectotherms, that the seahorse is going to be just as cold as the water. But the point is, these bacteria actually inhibit vibrio even if they're dead, so they don't even have to be alive to have an effect. So they've mixed these they've processed them and put them in foods, and they still have some kind of anti-vibriotic effect just because they already contain those substances in their body. So you don't need to be alive necessarily or growing. I would just say that if you really want to grow these bacteria in your tank, say like in a big pile of marine pure or some kind of biofilter like that, it wouldn't be as effective in the colder temperature, but it would be just as effective or almost as effective used in their food.
In the feed. I gotcha. Okay, real quick, I'm gonna ask the questions that people have asked real quick. It's gonna seem back and forth. Then I'm bringing in Holly and Cheryl. Guys, I do have to like literally leave the stream for 5 minutes to go pick up my kids. And if Kenneth has to go, he has to go. But it will only be 5 minutes. But real quick, Cora Works said krill oil is a really good source of astaxanthin, and you can mix it directly with rapashi type foods. No experience with. Was that just a comment? I guess that wasn't a question. I'm so sorry. And we talked about temperature. Okay, I'm going to go ahead and bring Holly and Cheryl in. I had other questions, but I know my daughters were getting ready to call, and I don't want to leave the stream for 5 minutes unprotected. I did ask about. We haven't discussed what yellow snow is. Can you tell us real quick what yellow snow is before I get the girls involved?
Yeah, that's so in a nutshell, PNS yellow snow is a product we developed after PNS ProBio. And it basically is just the purpose is to simulate marine snow. The reason for that is that marine snow, of course, is just an agglomerate of bacterial biomass. And it's a way to make, in this case, these bacteria, Rhodosudomonas, that nutrition available to filter feeders that can't harvest very fine particles. These bacteria are only like 2-3 microns long, whereas the particles of marine snow, or in this case yellow snow, are visible to the eye. And it's also really rich in B vitamins, so that's good for particularly vitamin B12, which most animals can't synthesize. No animals can synthesize themselves, and even very few not all bacteria can. So it has a whole B vitamin complex, and it actually yellow snow, because of the B vitamins, actually support the growth of the live bacteria we sell. So it's a companion product to PNS ProBio, if that makes sense.
And can you tell?
I was just going to say, so for seahorses, then I would say the uses for yellow snow might be to say mix it in with your Artemia or copepods or Daphne or whatever, and gut load them with these with the yellow snow. They do eat that. Yeah, and and then you can transfer the B vitamins and the trace elements and all the other good things that are in the yellow snow up the food chain to the fish that way. And then seahorse tanks also have a lot of gorgonians, and gorgonians in the wild love marine snow.
Love them.
To the extent that some of them are so specialized to exploit marine snow that they reject phytoplankton in favor of marine snow. So yeah, so being as a lot of gorgonians are used in seahorse tanks, I would say, there's probably a lot of cases where yellow snow would be good for that purpose, just to support the gorgonians in the tank.
And you did say you can't overdose the probio. Sorry, probio. Can you overdose the snow, yellow snow?
Yeah. So yellow snow is actually so that the bacteria they don't consume the whole particle and just eat everything up. They're pasteurized midway through the process at a stage where they're optimal nutrition, but they have they're large particle sizes and everything. So it's a non-live product, and you can treat that like any kind of food. Then basically, you can pollute the tank with it if you overfeed. Our recommended dose is pretty modest. So if you start out with that, you generally won't see, for example, an increase of nitrate or phosphate, but every system is different. So like with any food, you kind of fine tune based on your needs and the results.
Well, yeah, and organics, that's our big fear in seahorse tanks. But in a feed for the feed, in a feed that becomes the feed, that'd be a different situation. You could monitor it differently, I would think. And so since you can't overdose probio, and we're going to talk about the home grow kit, I really want to get into that. But how do you recommend using it in, say, a tank and in a feed? Is there instructions when you buy this product, or do you have instructions?
We do, but they're pretty pretty straightforward, again, because you can't overdose. So we don't really, people ask us about dosers all the time, for example, and I'm like, why make it complicated if you can't overdose it? Save the doser for something you can overdose, and just throw these in, every day when you're feeding with the food or in the food. And it's about as simple as that. We've tried to overdose to see what, even to get an LD50 or something like that. I've literally turned the water purple, adding so much and without any apparent harm to any fish or any kind of coral. So I'm pretty confident to say that, you'd have to spend a lot of money to overdose your tank with this stuff. In other words, you'd have to add gallons of it.
And just thinking, and there's the daughter, just a moment. And just thinking that using it in a feed, I'm thinking fry, I'm thinking live feeds, treats for the seahorses. It could improve their gut flora. I mean, it could prevent, oh my gosh, this is crazy cool. Tell us, I'm going to have to let Holly take over and run the questions while I go run and get my kids. But my last question for you before I come back is, tell us about the grow kit. Why can't we buy it, and what is it, and how cool is it, and why are you giving away $140 really cool thing that all of us want? Go ahead.
Sure.
Sorry.
So PNS Home Grow kind of came out of, so a lot of people use a lot of this stuff. People with big reef tanks or just people that feed it every day to their corals. And a lot of people started looking for a more economical way to use it. After they determined that they wanted to use it regularly, and asked if I had a bulk package or anything like that. And at that time I did not, but I was on a Discord discussion with some people one time, and they put forth the idea of PNS Home Grow. And I'm like, okay, I'm here to please the people. That's my job. So we developed a product, had to come up with a new recipe, in fact, so that it was all dry growth media ingredients, and basically made a package where you can grow these bacteria at home yourself. much in the same way you would phytoplankton, just using everything in the kit plus a few household items like bleach and hydrogen peroxide in a funnel. So the home grow kit is includes a 2.5 gallon collapsible container with a spigot, so that you can dispense it accurately and without a mess. And that also helps to prevent contamination of the culture vessel. It also, of course, includes the inoculum in a little bottle, and the culture medium, which is all just one powder thing. And it's as easy as making ramen. You throw the powder in, get boiling water, mix it up, funnel that into the cube after you've sterilized the cube with bleach water, and you let that cool. You add the inoculum, usually the next day. It takes a while to cool. And then it takes about 30 days to fully colonize the vessel. And then after that point, they last for 6 months. You just grow it on light, and we'd recommend you keep it on light. Any kind of house, even a 60-watt incandescent desk lamp, just some kind of source of moderate light. And you can just dispense it at will with a spigot, and you get 2.5 gallons. Treats at our recommended dosage. It treats 8,000 gallons, so it is truly a bulk package.
Wow.
Yeah.
Does the temperature affect that too? Like if you have it in a cold room, is that going to affect it?
Oh, that is such an awesome question. Yes, and thank you for that. I think that we've had very, very few people encounter any issues growing it in the past. So I think we did a pretty good job making it foolproof. But those few cases, in looking at them, we were usually able to narrow it down to temperature. So yeah, you want to keep it. We recommend trying to get it to around 83 degrees Fahrenheit. If you're using an incandescent bulb, that will help if the ambient temperature is warm enough. And in some cases, people have used inexpensive heat mats for like reptiles or seedlings. That you could just throw the cube on top of. So yeah,
I've had to use the chicken coop brood lamps for raising brine shrimp in the past because my house gets so cold in winter.
Is that like an infrared kind of lamp?
No, they're just high powered, like it's not even really per se a heat lamp. I think they're really bright. They're kind of a low grade heat, but people use them in chicken coops and stuff all the time.
Okay.
And I got them at Tractor Supply, and they're good for keeping my brine shrimp warm enough in the winter.
Yeah, that'd be great.
So basically, you're kind of in that kit. It's like making your own like PNS ProBio to some extent. Similar to that.
Similar. We were up until recently, we were cultivating the bacteria on an herbal extract. Actually, we since 5-6 seasons, I guess, since we started. Every summer we get a permit from the city of Boulder here in Colorado to harvest Eurasian water milfoil out of Boulder Creek. And Eurasian water milfoil is an invasive plant, obviously from Eurasia, and it's really destructive. So they always granted us the permit. What we do is we harvest the plant, and it has certain properties, certain tannins that these bacteria grow on. So they're actually grown on like this, like a tea, like full of polyphenols. And so telemagrandin 2 specifically is what this plant is rich in. It's actually it's an allelopathic substance. So it kills cyanobacteria and stuff like that. But these bacteria eat it for breakfast. They love it.
Wow.
So that's what we were using for a long time. It was a whole long process where we harvested it, brought it, had drying racks to dry it on, and all this stuff. And we moved towards a mineral medium, which is identical actually to the one that we use in PNS home grow. So we just get more consistent results that way. And also we don't have to be concerned about, a year where the city of Boulder doesn't grant us the permit, because it is It's a contentious thing because it's so invasive. They're so worried about people spreading fragments or whatever, letting things go downstream and all that.
Mhm.
So anybody who's used our product in the past, this is a good time to point that out. It has a certain look to it because it has that background coloration because of the tannins. If sometime in the near future you get a bottle and it's more straight red, but might look a little bit less dark, there's nothing wrong with the bottle. You probably actually have more bacterial cells in there than before, actually. But there's just the color's a little different because now it's we don't have that background color from the herbal color, all those polyphenols and so forth.
Interesting. So it's my understanding that PNS Bio ProBio can actually help fight high organics in your tank.
Oh yeah, absolutely. It's excellent for that purpose. For one of the reasons being
that's great for seahorses, I think, because our tanks, the way they eat and the way we feed them, and then the high organics, of course, cause the bad bacteria that causes them to get infection. So sounds like a great product for that.
Yeah. And they actually help eliminate, so they'll grow off of sources, like we were mentioning earlier, sources of carbon that other bacteria can't utilize, like cellulose and lignins and things like that, that just kind of sit around. Vibrio cannot use those either. However, if that gives these bacteria a competitive advantage over Vibrio, even if Vibrio can't use the cellulose, if these bacteria can, they will use that and proliferate at the expense of Vibrio for space and other nutrients. So that's
now since we tend to keep our tanks like such a lower temperature, like for instance, I have Erectus seahorses and I try to keep them below 72. They're usually in the high 60s. So how often, because I'm guessing because of the lower temperature, you might want to dose more often?
No.
Like what do you recommend for our needs for dosing them the water?
Yeah, so for bioremediation, I hate to say it, but Rhodosudomonas and the other PNS bacteria I know of would be useless in terms of bioremediation. They wouldn't be able to continue to grow, and instead of doing, sequester nitrate, phosphate, consume organics, because they can't grow at that temperature. So, however, as a probiotic, they certainly have use if they're administered directly to the animal gut through like a gut-loaded copepod or something like that.
So pretty much just for putting on their live food would be the really only beneficial.
Well,
for them, you think?
Yeah, or
at that temperature.
You can soak it into frozen foods and things like that too.
Yeah.
Okay. But, yeah, that would be better. Like in my case, my seahorses actually they don't really like live foods that much. I feed them pretty much
for it.
I gave them. Remember, I got live brine shrimp, Cheryl, because I was going to try to treat Scrappy Doo through live food.
Well, one thing
they looked at me like I was trying to feed them green beans or something. They're like,
one of the things too that happens with seahorses is they do not have a true stomach. Basically, they have a hollow tube that runs from their snout out to their anus, and I'm thinking that some of this stuff might be very effective. You were talking earlier about the product that aids in digestion of food and breaks down food. Something like that may prove beneficial to seahorses. Part of the problem we run into, I've started keeping seahorses around 2005, and so much has changed since then. And by the same token, there's still so much we really don't know about keeping them. And it makes it very difficult because even to this day, everything's, I consider a lot of stuff still purely experimental with seahorses. I was working with a research institute in Malaysia, and they got in 300 captive bred seahorses within a matter of days, all of them had developed tail rot. Well, again, that's a stress-induced, the stress-induced problem. And I was sending them, because they don't have the names, I was sending them actually formulas of different antibiotics and stuff. And the primary didn't want to try antibiotics and spend the money. They finally sent me a photograph, and yeah, they had tail rot. But the biggest thing is that they were seeing, is what they were thinking, the tails, they were infested with ciliates, and you could literally watch the ciliates consuming the tails. It's that kind of stuff that kind of makes you pull your hair out.
Yeah, for
Yeah,
sure. I mean,
it was
that
really-
many. Were they farmed? I'm assuming, or
I'm sorry?
they were farmed.
Yeah.
Yeah.
But it was one of those things. I even brought in another friend of mine who has a tremendous amount of experience with seahorses. They finally started listening to us. Within 3 days, they sent me another photograph. No more ciliates, no more tail rot. But that was just one of those things where I got lucky with what they sent me, and I was able to visualize what was going on. And that's one of the problems we run into, is if we can't get a good, clear picture of what's going wrong with the seahorse, it's next to impossible to give an accurate diagnosis.
Yeah, from what I understand of tail rot, there are many organisms that can cause that condition. So.
Oh yeah, yeah. And I mean, you can get anything from, it could be uronema, it could be any number of things. But it's not.
So is vibrio then not a problem at the temperatures that we're keeping them at, like high 60s, low 70, around 70?
Okay, wait a minute. What was your question?
I was asking about vibrio.
Yeah.
So at the temperature we're keeping, our rectus at around 70, and below, like I do, like high 60s, very low 70s at most. Is vibrio then not a problem really at that temperature?
The vibrio that we find in the Gulf, which is a little more, less on the eastern seaboard, is basically it proliferates above 74 degrees. And as it continues to proliferate, that's when it becomes deadly pathogenic.
Gotcha. So I'm wondering, would it be beneficial then to keep some of this, the PNS ProBio on hand to use if temperatures start to get too high in our tanks, as kind of like an emergency thing?
And I, guys, I'm back, and I've missed everything you discussed. I'm sorry, but why would we not be using this product as a probiotic, as we did Dan's probiotic, in a I can't think of the word, of course, but beforehand sense, like to prevent any problems if there were a temperature spike. Use it anyways, even though with all probiotics, we always say, well, you can't really see that it's working, but it is. And when a problem strikes, you don't have that problem, right? Thoughts on that?
Well, we were just discussing, though, it's only going to work in that sense if you put it in their food,
which is how we like to do it.
Unless your tank temps, are above the 74 that we keep them at. So I was talking about dosing the tank, and it's really not effective for dosing the tank because my tank temperature's too low. But I'm saying, would it be good to keep some on hand? Like, for instance, in the summer months, when it gets really hot, my chiller is fighting to keep the temperature at that 72 or below. If something happens and the temperature in my tank starts to rise, can I use this as an emergency treatment at that point?
I'll shut up and let Kenneth answer, but I would say also, I've been thinking a lot about this product since I started talking with Kenneth, and I think I might purchase it or maybe a grow kit for myself, because like when you're talking about fry, when you're talking, I mean, most of us seahorse keepers do give live feeds to our seahorses as a treat. It's not an everyday thing, but that makes it less expensive for us. But to put that in with the live feed and give that seahorse gut a boost of all the goodness, you know, he talks about the good. I just feel like that would be beneficial and make the seahorse immune system overall better, so that, all right, Kenneth, you answer, I'll hush. I got to go look at the wheel.
I'm not sure which question but
We
terms
do
of-
kind of bombard you with a lot. I'm sorry. Go on.
Kevin had the good blanket term there. I think prophylactic. So for prophylactic use, there could be some benefit to that, but because there doesn't seem, and I've never seen this with probiotics. I've never seen. It doesn't mean it isn't true, but I haven't seen it where probiotics are causing any kind of widespread resistance. It's generally when we add no. Yeah, so it's like, am I wrong, or you're concurring? No.
I've used probiotics for years, and I can truthfully say I've never had a problem with them.
Yeah, and it seems that all the problems seem to come from when people directly add them to the water, liberally and generally, to a system, that we get issues with.
I have a scale, and I know the exact water volume of each system, and I weigh out the amount that I use. It's just a little letter scale. I've got two of them actually, but and it's one of those things where that way I know I'm not overdosing or underdosing, and I'm consistent.
But in this case, with this product, it can't be overdosed, and apparently what we're discussing is that it's better used in feed, right?
Potentially. I mean, I have to admit, I'm only guessing there, but it's certainly a more natural means of doing it, and it seems, just from what I know, like I've never seen where probiotics are, you know, if we're worried about resistance, that doesn't really ever seem to be an issue with probiotics. But in terms of like having it on hand for an emergency, that's certainly feasible, because, you know, the bottle, by the way, is stored at room temp. So it's really shelf stable at really cold temperatures. We just did a test to see if they withstand freezing. So say you get a bottle in the mail in the winter and it's frozen solid, they're actually completely viable in very short time upon thawing, which is cool. We've also done studies with extreme heat, and found that it was over 130 extended exposure of temperatures over 130 degrees, which is more than you typically encounter in a hot day shipping, even if it was left of a FedEx truck or something like that. Again, perfectly viable. It's not a surprise there, because these bacteria love heat. But anyway, yeah, very shelf stable. You don't have to refrigerate it. Just let it, keep it on hand, and it'll last as much as 8 months just sitting there. So it would be ready if you wanted to keep it on hand for some kind of emergency. Specifically,
nice. How long would you soak food in it? Like if I'm going to use it for my frozen mice,
or is it better used with live feeds?
How long would you soak?
Well, yeah, so that's more of an art than a science for sure. I'm sure it depends on the food you're using, because a lot of these things require different kinds of binders and so forth. You may put the food in the water, and especially if the animal doesn't consume that food item immediately, most of it's just kind of washed off, unfortunately. So that would definitely depend on the food. If they're eating something like a pellet that you can soak, that would be much more effective, because you wouldn't see as much of that washing away effect when you first put it into the aquarium. I would definitely say gut loading would be a more effective way of delivering the highest number of cells.
Guys, I am back, and I am doing the wheel. That's why I keep disappearing. I will be back. And Kenneth, I'm sorry we keep bombarding you with questions. Go ahead, Cheryl.
Well, that's half the fun of it. I haven't heard anybody use the term LD50 in probably 15, 20 years. We used to do it all the time. So, do you
Cool
know
and
what
interesting.
LD50 is, Holly?
No.
It stands for lethal dose 50. What that is the amount that you have to use of a substance to create a 50% mortality in the population. It's a
Oh,
scientific
jeez. That's morbid.
That's a scientific term.
So, Cheryl, I'll ask you this. So, if you were using this to soak the frozen mysis before you feed it, how long do you think you'd have the frozen mysis in it for it to absorb it? Do you have any idea?
I really don't.
Yeah, that's a
It,
tough one.
it's
Because I know I've used the powder stuff,
yeah. The thing is with
you
probiotic,
know, the vibrants, and I mean, I never knew really how long to do that. They said till they turned red or something. Could that be a good guide, maybe?
yeah,
I
You know,
personally never liked
It's a
soaking.
problem for
I've
me.
never liked
Yeah,
soaking frozen foods because it instantly comes off the second you put it in. However, in this
yeah, that's
case,
even
but in
thinking.
this case, I don't think it would matter because you
yeah.
can't overdose the tank.
It doesn't
It
hurt
doesn't hurt
anything. Yeah,
anything.
you're not polluting
I
the
mean,
tank, so I don't see any reason to worry
nobody
about
want.
that.
And no, it's a good question though. I mean, if you want to get it into the gut of the seahorse, that's a great question. But I
Yeah,
just think I would focus more on using it with live feeds, and we do, with Artemia treats or
I would
fry.
if they would eat them. But I was explaining while
Oh,
you were
sorry.
gone because I tried to give mine the brine shrimp.
Yep.
Remember, I got live adult brine shrimp for them. Most of them didn't want anything to do
Right.
with
Right.
it. So the only thing they would eat live so far is the
Right. I was
mysids.
gonna
They
say
like to
though, I was gonna
cook.
say you can feed the Artemia, you can feed the probio to the Artemia, feed the Artemia to the mysids,
Mhm.
and then feed the mysids to the seahorses. And that's a lot of digestion that's going on. I don't have the scientific proof to
well,
say it stays in
let,
there, but
let
I
me
bet it does.
let me go back again. You were talking earlier about actually enhancing the digestion in the product. Okay. Tell me a little bit more about that because one of the problems we have with seahorses is they do not have a true stomach.
Mhm.
Basically, they're a hollow tube. It's not quite that bad, but it is. And which means you can actually watch them pass live food that they have ingested, particularly in young fry.
Yeah, certainly all the chitin and stuff like that. There's no way that. Yeah, so these bacteria are normally they occur in the guts of all kinds of animals, even terrestrial animals, especially herbivores, because of their ability to digest cellulose.
Right.
So rhodospirillum, one of the species we culture, I see a lot in gut flora, even weird things like wolves and stuff. I'm not sure how they're getting it unless they're getting it from the animals they're eating. And but yeah. So what would happen is these bacteria, so long as they're active in the gut, and we do have evidence through simulated experiments in a simulated condition of the gut with the appropriate pHs and all that, that suggests that these bacteria survive passage through the GI tract. So most
I'm always curious about stuff like this because it particularly is common, particularly in newborn seahorse fry, where you can literally watch them ingest and pass live food.
Mhm.
And that's not doing them a darn bit of good.
Right. Yeah. Most I'm sure most of the little crustaceans and stuff that are enveloped in a bunch of chitin and everything it's basically it literally goes right through them.
Yeah,
completely.
that's why I pretty much rely on Apanamensis copepods for most of it, because the brine shrimp, particularly if you do it at the wrong stage, it's going to go right through them in a heartbeat.
Yeah. So,
but again, this is a product that won't overdose a tank, so it's not hurting anything. It
Exactly.
can't hurt anything, and it may very well be gut loading the seahorse with fighting things that can fight vibrio and everything else. Go on, Kenneth.
In terms of nutrition, I mean, direct nutrition to the animal, if they're in a food and they're properly bound to even to like a frozen food or something, the bacteria themselves are incredibly nutritious. So it varies depending on the growth conditions, but often they're over 70% protein. They have the B vitamins, vitamin E, obviously the carotenoids, which have different health benefits, particularly as antioxidants. But as opposed to like, say, phytoplankton or something like that, they're very highly digestible because unlike a phytoplankton or any other plant, they don't have that cell wall. So even a young seahorse, I mean, they're just like, I'm sure that something like that, especially with all those little cells that are just a couple microns a piece. And so in other words, so that the digestive enzymes in the animal gut could attack each particle from, I'm sure the bacteria themselves are very easily and very quickly digested, so that, especially in the case of a juvenile seahorse, that might be really beneficial, even as a direct feed.
Yeah, I'm thinking because when I hear about new stuff that I haven't heard of before, my first thought is, I got to try this out.
Yeah, and as opposed to some other, you mentioned powdered probiotics. Now, those are sporiform bacteria.
Right.
That's usually bacillus and stuff like that, which they're proven value as probiotics. But when they're in the form of a spore like that, the benefit is limited because the animal will pass the spore before the organism even wakes up and starts doing anything. Especially in the case of a seahorse, where they might eat something and it goes right out the other end in very short time.
Well, that happens quite a bit. In fact, for a long time, people were swearing up and down that baby seahorses were ingesting air and getting it stuck, and then they were floating. Well, we have long since doing microscopic work. That's not the case. That's not what's going on with them. And in fact, it was totally off. What's causing the increased in the swim bladder, the appearance of the swim bladder, is actually due to an inflammation in the GI tract due to poor quality food.
Which this product could help. I mean, seriously, like I don't mean to keep going back to the product, but I mean, Holly, everything we've talked about in the past about how seahorses just all of a sudden have liver problems and all these problems. If you were able to gut load them with probiotics with this product, I mean, I'm going to try it myself personally.
Yeah, no, I want to try it. I'm just trying to figure out how to use it so they'll absorb it. Because like I said, the only thing they'll eat so far that's live is the myces. And they cost a fortune.
Right. No, I hear you on that, but I will get with Dan to try to get, because he had that down to a science, the conversion rate. What, how much you had to feed the Artemia, to feed the mysids, to feed the seahorses. My seahorses will eat Artemia, so I don't know.
Yeah, I have a few that did. It was funny because certain ones liked them, like most of the girls. More so than, but some of them were looking at me like I just tried to give them green beans or something.
Get their nose and tell them this is for your own good to eat this. Like raising kids.
All right, you guys. So Kenneth was kind enough. I wanted to steal it for myself, but I thought, no, better not. He was kind enough to give away one of the home grow kits. And can you recap again what comes with it, Kenneth?
Yeah, we went over that a little bit while you were gone, but
sorry.
That's okay. It's basically everything you need to cultivate these bacteria at home, with just a few things like the to clean it with bleach and all that in a light and in a warm spot.
And I heard you say peroxide too. You're talking our language. It's bleach, peroxide. That stuff works. But
yeah.
anyhow,
So you guys all
okay.
have that on hand.
You know we do.
Yeah.
And we're the type of people that will cultivate and use this. And I could see one of us, whoever wins this prize, if you're in the Seahorse community, please cultivate and talk
I
to me.
volunteer.
No, you can't volunteer. You can win though. All right, let's see if I can share the wheel real quick. And we didn't give it much time, but that just goes to show that if you pay attention, you get invited and you get to win. All right, here we go. I need to put music to this. I keep saying I will. No way, Holly. I did not. There's no way for me to rig this. No way. All right. Okay, Holly, you won it. So
awesome.
So we will.
Yeah. See, now I get to try
Okay,
this stuff.
I want to rerun.
No, I have no way to rig it. It's internet. But Holly, I know you will post and show us how you use it, what you use it for, how it works out, and do posts and share with not only Seahorse Whisperer page, but also Hydrospace page and show. There's some fantastic articles about people that have used this already. And
if you need pictures, send me some and I'll take pictures and send them back to you.
There you go. All right.
See, I'm wheeling and dealing now.
So I have a question about it though. So is it the same as the PNS Probio? I forget what you could, the Roto.
Yeah.
So she has 3 different species, right?
Strain species. Yeah.
That's an important. Yeah. So you select one of 3 species. So that would be either Rotopseudomonas palustris, which is a species in Probio, or Rotopseudomonas and then Rotospirillum rubrum additionally, which is additionally in substrate sauce that we mentioned, the cycling product. You can get Rotospirillum individually, or you can get Rotobacter, which actually Rotobacter is an interesting one too in terms of treating vibrio. There's a lot in the literature about, it's all pretty mysterious how it works, but that one definitely also inhibits vibrio.
Yeah, but will it help uronema?
Wow.
I don't think I've never seen where it helps us. Anything like that. It kills protozoans or
one of the species that I've worked with and I love dearly has a history of being carriers of uronema. They're coming out of Asia. And
so we need you to do more work, Kenneth. We need you to do
now
more.
I have to start working with ciliates.
That's right. We need ciliate help.
Yeah.
But go on with what you were saying. What were you saying about the 3 species? Because can they all be used in marine aquaria?
Yeah, I would say the first 2 are more likely for marine if you want to use it specifically for bioremediation. In other words, if you want it to survive and reproduce inside the system. The Rotobacter we have, unbelievably, I haven't tested it in saltwater yet. Partly because we initially misidentified it as capsulatus, which is a freshwater species. But then after we had it analyzed, turns out it's Rotobacter spheroides, which is euryhaline. So it could live in saltwater and definitely does. So if that's the case, that one too could live in a marine system, but we're not as sure about that third one, Rotobacter. So I would probably recommend for first time growing it and using it for this particular application as a probiotic. I would probably just recommend Rotopseudomonas.
That's what I'm thinking, but it occurred to me because we're talking about the seahorses, but I have a freshwater tank also. And that one I keep around 75 and above, 78. In the summer it gets to like 80 or so. So I can start adding it to that and see how it helps
Now,
Yeah.
does
them.
the species
'Cause you
matter
said it's
though?
fresh and salt.
So
water.
No, I would
they
point
are
out
fresh.
that all three of them live in fresh water.
Okay.
Yeah.
Yeah, I remember you mentioning that. So I'm like, oh, I could put that in the freshwater tank.
So we're gonna get tons of results from you, Holly. Good deal.
Yeah.
It does make
yeah.
8,000 gallons, so anyone in your area,
Use it in every tank.
you'll have opportunity to share.
Maybe the goldfish will even start
We'll
getting
be hitting
some.
Holly up, but then we'll be buying it because it turns out to work. And Kenneth, did your wife ever get me the video of the walkthrough?
Oh, she sent it to your
Email?
Google Doc.
Okay, did she share it with me?
Yeah, she said she did.
All right. I just wanted to make sure I got to do that if
Yeah,
that was the case.
that basically just shows how we make it, the facility. I'd be down there today, but the reception is so bad. That it sometimes cuts out and gets junky. So the video is going to be much better.
That's what you get for living in Boulder.
Well, now we live out of Boulder County now. Even I live up in Johnstown, if you're familiar with the area. But
Oh yeah, I used to live up at Estes Park for a number of years.
very nice, very nice.
Yeah, it was the only catch is in the wintertime when you have to half mile in to get to the front door because the car's not going any farther.
Right. Yeah, that's up there. I used to live, I don't know if you know, like west of Lyons. Yeah, I lived in the mountains out west of Lyons on Highway 7, out by Peak to Peak Highway.
Yeah.
Mountain stuff. And that's where I was first developing these bacteria.
Well,
Like
I got my bachelor's from up in Fort Collins.
Oh, okay.
Yeah.
CSU?
Yep, CSU.
Okay.
And then I went to UC Boulder for the master's portion.
Nice. Wow.
It was one of
And
those
what
cool,
program?
it was one of those cool things where, hey, I got to spend half my life running around in the woods.
It's beautiful out here.
Yeah.
I miss living by the ocean. I've lived on both coasts and I'm in just about everywhere else, and I'm stuck here now for family, and the house we just bought, of course. But Colorado's pretty nice. I like it. We've got a good.
It gets cold in the winter, and it is prone to fires. But that's The other thing you got to watch is depending on where I used to be an avid backpacker when I lived there, and you got to definitely watch out for the wildlife.
I can't imagine Cheryl backpacking around everywhere. That just makes me. That's so crazy. Okay,
I was actually got caught up in Rocky Mountain National Park, and I heard a black bear roar.
Ooh.
And I knew something was up. Next thing I do is I see two cubs running by.
Oh, and that's danger.
Well, I jumped into a ravine. The only problem was it took me about 45 minutes to get my happy buns back out of that hole I got myself into.
Yeah.
Now, one of my better ideas.
Well, go ahead. Sorry.
I was going to say, those are the things you do when you're young and foolish.
Absolutely. Well, I'm old and foolish, and guys, I can't find the video that Kenneth has sent me. So all that means is we're going to have to do an actual post and maybe share it at the beginning of next week's live because I can't find it, Kenneth. I'm sorry if she sent it to me.
Another alternative is I could do a live walkthrough. It just wouldn't be the best quality, but I could walk right down and
go. Well, let's try it. Let's try it.
I'm all charged up here, so
all right.
Is there a way I can flip the screen? Yeah. Do I just click on my window?
Let's see. Let's test it out. No, not working. Okay, Holly, you've flipped your screen before. How does he do it?
Well, on my phone, are you using an iPhone?
I'm on my laptop, but I can kind of just
maybe go to your camera settings.
Okay, where's that?
Holly, give him your phone.
Settings.
Give him your phone.
Yeah, give him your phone. Yeah.
camera setting. So Kelly's pull down here is blocking, I think. It's down over some of the icons below the screen. I don't know if the setting one.
But you have to go into.
Is the setting one?
I think you have to go into settings. Go into settings in video.
Oh, is that the wheel one?
Yes.
Okay.
Yeah, it'll be
Click
the
that
wheel.
guy once. Front camera. Ah, I see.
There you go.
Sorry,
Cool.
guys. We'll figure it out. We want the walkthrough.
Ooh, I could have a virtual background.
Yes, you could.
Audio.
It looks like you're a musician also. Do you play guitar and stuff?
Some of those are mine,
Or
and
some-
some of those are my wife's. She actually teaches ukulele, taught ukulele, and we even just got a piano,
cool.
which I don't play at all. I bought it right after I came back from reef stock to take up the last remaining space where I could buy an aquarium.
Just stop yourself.
I was too tempted to buy all the corals while I was there, and I figured the few $100s it cost to move the piano would cost far less than what I would eventually, the $1,000 I'd invest in
What you
corals. So yeah, it was a safety
Absolutely.
measure.
Good strategy.
I'm just gonna walk on down and
Please.
hold the camera out, and we'll see what I can show this way.
All right.
So now you're seeing a back view of me walking into the domain, and I'll start in let me hit the light too. What we call the kitchen, and that is the area where we do all the actual brewing. So I'll hit a couple more lights here, and so what we see here. So it's the brew kettle and the hood and all that stuff. So obviously we do everything in really small batches. So that helps with quality control and to just maintain good biosecurity and everything. We drop those into these cubes. You'll see a lot more of these, but these are identical to the cubes that are in the kit, except they're 5 gallons. So they're twice as big. Our production size cubes, but a lot of action happens in here. I spent more time than anywhere else standing in front of the sink. Unfortunately, cleaning things just requires a lot of that.
I feel like that myself with the Seahorse.
Yep. And so we have this table is basically where we do all our bottling and the labeling and all that. But the computer screen is kind of an additional. We hook that up to the microscope, so that's an additional level of biosecurity. So we check over the cultures, make sure everything looks good, and
I love that.
Do some photography. But yeah, it's just all the packaging materials and so forth are at hand, bottles and so forth. This is like I just reordered bottles today. That's the minimum stock we keep. It's it takes up a lot of space, but we got everything in one place now, and it's nice that it kind of has a working order. It just goes from the kitchen to packaging, or if we're brewing, it goes from I should say when we're bottling, if we're brewing, we get these cubes and we roll them in these carts all the way over to the grower area or whatever, which is over here. So these are the mothers basically where we select. I used to take the really good cultures and then use those for product, which turned out to be really dumb. You want to save your good product and use that as mothers. So all of our product is made with product. So it's kind of another level of quality control. We always know where everything came from, and we know that our best cultures are being used to make the new cultures.
And you can stand behind them. I love that. Go on. Sorry.
Mm-hmm. So our inventory right now is incredibly low because we're phasing out the old formula I was mentioning earlier. We cultivated on a herbal extract from an invasive plant, but now we're moving to something that is more consistent. The mineral medium, which is basically the same thing you make PNS home grow the kits with. So it's kind of hard to maneuver going backwards, reverse, but They're just grown on these racks because the bacteria are photosynthetic. It's all on these lights, LED lighting. We used quite a bit of juice to grow these guys. So I actually had to put a sub panel in to handle all the extra electricity. That, and also because of the heaters. So, we run these heaters all the time to maintain a temperature of 83 degrees Fahrenheit down here. So it's always really toasty. We can't really heat the individual cubes effectively, so we just have to bring up the room temperature for that. This is kind of where all the action is. Test tank, which is getting converted into a warm water tank now. The corals we used to test aren't doing so well at 83 degrees or warmer sometimes, so I'm going to turn that into a freshwater tank with some discus or something that like really warm water. But here is a good example of a mix of things. You see all the different colors because it is different species at different stages of growth. So if I can really fix it, this guy, that's Rhodopseudomonas. They always look different in the camera, but in person it's kind of a peachy pink orange color.
Ooh, they look so brilliant.
This is what they actually look like when they come out here. Just absolutely clear like this. And then after they're inoculated, they grow out a little bit, and then you start getting a little pinkish color, and then they go into like this. This is Rhodospirillum. The other one, this is my favorite. This has got a really pretty, in person, it's almost like a magenta color. And then this is Rhodobacter, which, believe it or not, when you shake it, it really looks yellow here. It looks orange to my eye. And if I were to stir this up, it would be more red. But Rhodobacter really likes making films, and then those eventually just kind of fall off and settle on the bottom into like a sediment. So it just
That's awesome, and it looks so yellow to us. Go on.
Yeah, it's the lighting's always weird in person compared to the camera, and then of course the lights are so incredibly bright. But they all different species here. These all look different to my eye, and they all just kind of look orange here. But different ways that the carotenoids are absorbing and reflecting light, which is what carotenoids do. And here are some really young ones. I don't know if you'll see if they look pink at all to you, but that's kind of the early stage. Yeah. So for anyone growing the home grow, that's the first thing you're going to see is like this pinkish color, and then it just progresses to like this Rhodopseudomonas, like you get the orange and then the reddish, like a fiery red orange.
That's amazing.
Yeah.
Very cool. Where do you find those containers?
We get them from a supplier. Same place we get our bottles. So it's all, it's all brought in by like motor freight. We buy them in huge quantity, or else they're actually not very cheap unless you buy a lot of them. So that's what we do, and then we have to store lots of them. It's just
doesn't one come with the home grow kit?
Yes, they come. Yeah, the grow kit. So that would be like the grow kit is this little teeny 2.5 gallon. So it's half the size of the ones you see in our production area here.
Okay.
So the 2.5 gallon one it can treat 8,000 gallons. So that's certainly enough.
That's enough. Yeah.
The 5 gallon ones are a little less manageable for people because they're quite heavy. I think they're over 50 pounds and kind of hard to hold on to because it's like a collapsible cube, so it's kind of soft, hard to pick up.
They don't have a handle either.
No. So
yeah.
So the 2.5 gallon was a nice balance in terms of being wieldy,
doable,
and also producing the bulk amount of product that people were asking for. But this is basically it. Not anything complicated. That's why you can do it at home. You just brew your culture medium and put it in the cube and put it on the lights and
keep it warm and lit.
Keep it warm. Wait 30 days and you're golden.
So I'm curious though, and I've asked Dan this too, because he's come in and explained things to do with seahorses, and you've explained how to do this with bacteria, and you sell a grow kit. doesn't that hurt your business?
Maybe, but I enjoy what I'm doing and I like sharing it with people. And, as long as I'm able to fill my fridge doing what I do, I'm happy. A lot of people support it. Yeah. A lot of people have been supporting us, and that's great. If other people start growing it, or even if I end up encouraging a competitor out there to, so be it. That's how business works. But, I do what I like doing. I've been in this industry for over 30 years, and 20 of that was just working in live feed. So it's there are a million other things I could think of to do if I had to, but I'm just focusing on what I love doing, I guess.
Well,
Sharing it with people is certainly one of the most fun parts of that.
Well, I'm excited to try it out. I can't wait to get it started.
And I would say that Dan too always said that too. Dan always said that too, is that sharing the knowledge and helping everyone in the aquatic industry succeed was the most beneficial thing for him, and obviously for you. So I just thank you for not only sharing all this information with us, but also selling a grow kit that can allow people to do what you're doing. And then, when they get sick of it, like me and Holly always will, then they just buy from you.
Kelly,
But
Kelly, did you see this? I thought it wasn't going to say it, but Kevin said it anyway. So, has the police showed up at your inspect your meth lab?
Yeah.
I've got it's legal to grow marijuana where I live, but it's funny, we have the lights, the light like pouring out of the basement, like 24/7, and like I said, I had to put in a sub panel because we're consuming ridiculous amounts of electricity. I'm surprised I haven't had the DEA knocking at the door. And all the weird chemicals I order, and
a friend of mine who actually lives in the Netherlands, and he raises dwarf seahorses. He has been doing it for many, many years. Well, one night, law enforcement with a dozen officers show up because they're seeing the purple-colored, blue-purple lights. So what does he do? Now, you have to understand, this guy has got a heart of gold, and he's very helpful. He invites them all in, and this is teaching them how to raise dwarfs, and ended up selling some of his dwarfs to some of the officers. And so,
Love it.
Oh, great salesman too.
Right.
Yeah.
But I thought that was that's one of my favorites. Now I've got another one for good people intervening.
Yeah. Well, I am interested. I can't wait to see Holly try this out. Can't believe you won. Honestly, that was just a fluke.
Yeah.
But
it's sweet. Thank you, Ken. I can't wait.
Thank you, and congratulations.
And I did promise Ken I wouldn't keep him over 2 hours. I promised him 1 hour, but hey, it's two.
Is it? It never feels like
I
that
know.
long.
We could talk all night. I know. And I love you guys
They
in the
really
comments.
can. They really can all night.
We can. But can I?
Well, it's a lot earlier here in California.
Right. See, for me, it's 10 o'clock, guys. I got to work tomorrow. But I would say I am going to either publish Kenneth's notes or do an article about it, probably both. And we will definitely do a recap video of this. And if you're interested in never coming back, I would love to have you and Dan talk about probiotics and all this stuff. He's not available right now. He's having some medical issues, but I would love to see you guys have a conversation. It would be so amazing. So you're invited back always, every week, but we'll try to get you back on as a special guest when we can have some of these other people here. But I just thank you so much for coming. And I'm so excited to learn more about the products. And my gosh, suppressed vibrio. Holy shit.
Holly, pictures are mandatory.
Yes.
Oh, well, me in pictures. I'll
get a
get plenty
tag.
of
She's
pictures
great.
for you. I'll probably have plenty of questions too, Ken.
I'm here for.
That's the
Okay.
perfect outcome then.
I'll be
Exactly.
I might
And
too. I'll
You
be
got-
honest. A lot of people, we're all doing this together. You'll see in the notes, a lot of the papers I cited were published like within the last 3 years.
right.
This is
It's
new
all
stuff,
new.
frontier stuff. So we're all learning together, even the scientific community. So yeah, certainly I'll be asking questions, asking how things are going with you, and that'll be interesting to see, as
And
it is with everybody.
And
How-
again, sorry for my silly questions, but I'm not a scientific person. But I mean, you help explain it very well and help me understand it. And that's what needs to happen for aquaculture to take off, every level. And so I just thank you. And with that, guys, was there any last minute stuff? I am going to have to end it. I'm sorry. I'd love to.
Yeah, there is a
Okay, go
question
ahead.
I see from Coral
right, let's see.
Works. Do
I'm
you see
looking.
it?
I'm sorry if I missed your questions, guys. I was gone.
Right after Kevin said goodnight, or before Kevin said goodnight, it says, how sterile do y'all have to keep your phyto and copepod and whatnot cultures? I usually see glass jars recommended, but in freshwater, there's a lot of plastic bucket culturing for infusoria and scuds, etc.
Good question.
Oh, holy cow! Yeah,
I've grown copepods in buckets myself.
yeah. So for growing bacteria, it's a whole other game. Like
Yep.
when I'm transferring cultures, I hold my breath. Anything can contaminate these, so that's why we rinse out the container with bleach for the home grow. You'll want to rinse that out, and then we don't even rinse the bleach out with tap water then, because the tap water has spores in it that could potentially infect. So we
Absolutely.
shake out as much of that bleach water and pour it in at above boiling. We pretty much give zero chance for any of these things to contaminate. But one fun little story, I guess, before we close, and just to address that particular issue, when I was my first microbiology class in college, in the first lab, what they had us do is they gave us a pile of petri dishes and had us walk around to various places in the building. Maybe on the spot, we'd go to the bathroom. One was like right outside, and open the petri dish. I think we had some had to be open for like 1 second, some for 30 seconds. And then we came back and looked at everything. And of course,
Oh my gosh, that's
this-
going to be scary.
And you never wanted to go back there
Right.
again.
no, especially the bathroom. But yeah, it was everything was contaminated, most notably including the ones that we had just opened and closed. So they were only exposed to the air for a moment. And of course, because of all the airborne spores and things like that, it still managed to get contaminated. So for CoralWorks question, I would just say that you're always, unless you're working under a hood and you have like, they even pump air out of the room in a certain way to keep the airflow away from a negative pressure thing. Or it's like, unless you're working in a lab that's specialized for this kind of thing and you're working with really small amounts of cultures and everything, you're going to contaminate, and I contaminate every day, everything. It's the game is to keep it as low as possible.
Yep.
So when you're working with bacteria like this, or even phytoplankton in some cases, you definitely want to make sure all your instruments are clean, your hands are clean, if possible, wear sterilized gloves. And if you're like me and you're super paranoid, you even hold your breath when you are working over an open container.
We say just with keeping seahorses, make sure that you have separate nets, separate feeding dishes, separate everything, because the airborne, as you said, you can't even control. It's going to happen, but you can control everything else. You can try. You can do what you can to make sure biosecurity is the best you can do.
100% sterility really does not last long as a general.
You can't. I mean, you can't. It's
I mean,
impossible.
Yeah.
You can buy all kinds of antibiotics and all kinds of stuff and culture systems and everything else, and you're still going to contaminate it.
That's
gonna-
why I love the idea of probiotics. Hit the problem before it happens. You know what I mean? I mean, because you know what's going to happen. So hit it first. I don't know. I like the idea of that. And
My favorite culture was Tetrasilmus years ago in a 5-gallon glass jar, and I kept it going for over a year in that jar. I was so proud of myself. That was my first attempt.
Kevin said he does phyto in clear plastic containers and rotifers in food grade 5-gallon buckets. Clean and simple or sterile, sorry, is important for phyto. Rotifers are easy. That's true. It depends on what you're culturing as to how much you can contaminate it. With bacteria, though, that's a whole other level. Like, I mean,
I can imagine.
That's an interesting thing too. Now, like you were saying, as they say, life will find a way. Things will always get into even our live food cultures.
Yeah.
And I wish I could have cited it. I couldn't find it, or I didn't think to look. But I've seen somewhere where vibrio can infect Artemia cultures. I think you can
No
even
doubt.
That's not surprising.
Undecapsulated cysts or whatever. But often when we're feeding like phyto pastes and things like that are messy and create, you're certainly going to have a lot of background biology in the live food culture. Some of it, which may be potentially pathogenic. So it may be good to have a probiotic, and specifically an antivibriotic probiotic for that purpose.
Absolutely.
Vibrio is pretty darn nasty. I did a paper on human lesions with vibrio, and I'll tell you, those things, I mean, you're talking months and months of treatment, excruciating pain, and they just never want to heal. And eventually, you're at it for a long.
Are you talking about seahorses?
People.
Or people. Oh, okay, sorry.
No, humans.
Okay, sorry.
They were actually seahorse keepers that got cuts and then started handling their tanks. And the thing is, they didn't realize what this was back when we were first learning about vibrio and cuts and seahorses and all, and they did not realize that they were contaminating themselves, and they had the presence of mind to help because they didn't want other people to do it, getting the same problems of sending me all the photographs and everything else and the stuff from their doctor. And I put it all together. It's all on seahorse.org.
Yep. No, I hear you. No, I just wondered because I was able to successfully treat seahorses for vibrio, but that's again why I'm so excited for a probiotic that has these antibiotic properties that can possibly gut load these seahorses and making them less susceptible to the problems. Kevin said I did a rotifer harvest once, and then in phytoculture, and then I had rotifer contamination in phyto, and I had 10 million rotifers. Right. I've talked with Chad from oh my gosh, Reef Nutrition. Yeah. And he talks all the time about when you try to do it yourself, and if you don't take care about biosecurity, you end up with copepods being eaten by rotifers by, it's a dang mess. So, and with bacteria.
They can't grow a rotifer to save their life, and then they try growing phytoplankton. Then they go, I'm going to grow live phytoplankton, and then they can't keep. Then the rotifers go gangbuster in the phytoplankton.
Yep, it's insane.
Hey, I bought a bunch of rotifers from what was then Reef Nutrition, Reef Mariculture. Anyway, I had to call back a week later for something else, and I told the guy on the phone. I said, "Oh, by the way, you were supposed to send me 1 million rotifers, and I was 1,001 short." So he's getting ready to send me a whole new shipment. I've got four or five containers of rotifers already set up. I don't need any more rotifers. I was totally kidding with him. And when he realized what I was doing, he just burst out laughing. He goes, "You got me good on that one."
All right, you guys. Okay, so I am going to have to end it for tonight. Kenneth, thank you again for coming and showing us your facility, sharing your new products that, not new, but new to us in the seahorse world that can possibly help us. Holly, we expect many pictures, videos, and
Yes.
updates to tell you how it's going. And Kenneth, again, we invite you back every week, but I will set up a time for you to be here when Dan and Ray can too, and we can have this
Yeah.
really amazing conversation about biosecurity and bacteria and probiotics and everything. And any questions that I did miss, I'm sorry, you guys. I will post the notes that Kenneth sent me, and I will post a recap of this video. So
I love this and thoroughly enjoy learning new things and seeing new experiments and successes coming out.
we need more people like you,
Absolutely.
Kenneth, making family businesses that actually help not only the industry, but every
Come back,
industry.
we'll get you going on raising seahorses.
Yes. Right, we need to get
Yeah.
some seahorses.
I just put a piano in the last spot I have.
No, we're gonna
No more of that nonsense.
we're gonna talk. I'm gonna get to know your wife. We're gonna move that piano, have a seahorse tank. No, I'm just kidding. But I will also post that was the other thing, is that I will post the walkthrough that was sent to me that I couldn't find. I'll get with Kenneth afterwards and I post that so you guys can see it. Even though he showed us, I just still want to post the video. So again, Kenneth, thank you for coming. Thank you for staying so long. I know we talk all night, but it's fascinating information, and we're very excited for everything you're doing, everything you've done, and will do in the future. So thank you for coming, and we hope you come back.
Thank you. I will. And it was my pleasure. Have a great night, everybody.
All right, everybody, let's do a cheers. I'm sorry, I gotta work tomorrow.
Cheers.
All right, happy Wine Wednesday. Cheers.